In 2015, a geneticist pulled a 5,000-year-old tooth from a bag and found something that should not exist. Inside that tooth was DNA from the deadliest organism in human history, Yersinia pestis, the single deadliest bacterium in human history, the organism that caused the Black Death. But this tooth was not from medieval Europe. It was from a Neolithic farmer who died 3,000 years before the Black Death. The plague did not begin in the 14th century. It began at the dawn of civilization. And what it did to our DNA over 5 millennia is the most terrifying discovery ancient genetics has ever produced. Before we go further, understand what ancient DNA can do. A single tooth can preserve the genome of every pathogen that was in your blood when you died. The dental pulp chamber is sealed from the outside, protected from contamination by enamel and dentin. Whatever killed you is locked inside. The story begins on the Bronze Age step. In the early 2010s, ancient DNA labs across Europe began sequencing teeth from burial sites in Russia, Scandinavia, and Central Asia. They were looking for ancient migration patterns. What they found instead was a deadly pathogen. In 2015, a team led by Simon Rasmussen published the first detection of ancient plague DNA in Bronze Age teeth from the Afanasevo culture. The bacteria had survived inside the dental pulp for 5,000 years. Teeth are time capsules for bloodborne infections. Whatever was in your bloodstream when you died gets trapped inside the pulp. And what they found trapped inside these teeth rewrote the entire history of plague. The ancient strain was missing key genes that would appear in later versions. It lacked the ability to survive in flea guts, meaning it could not spread through the rat flea cycle that powered the Black Death. Instead, this plague spread pneumonically, lung to lung, breath to breath. In the cramped longhouses of early farming villages, one infected person coughing through the night could infect everyone sleeping beside them. And the archaeological record shows exactly what happened next. Across Northern Europe, entire farming communities vanished within a few generations. The Tripilia culture of modern Ukraine, one of the largest proto-urban societies on Earth, collapsed. Settlements that once held thousands of people were abandoned and burned. The first plague did not need rats. It needed roofs. And farming gave it exactly that. The transition from hunting to settled farming created the first pandemic. Dense populations, shared air, grain stores attracting rodents. Since 2015, plague DNA has been found in Neolithic and Bronze Age teeth across the continent. Sweden, Latvia, Germany, Croatia. Each detection pushed the timeline further back. Each new site expanded the geographic range. One specimen from a Swedish burial dated to nearly 5,000 BCE. The oldest confirmed plague victim in human history, a young woman buried in a megalithic passage grave. She was roughly 20 when she died. She lived 2,000 years before the pyramids were built. And she died of plague. The phylogenetic tree showed that all later plague strains descended from these ancient Neolithic lineages. The Black Death was not a new disease. It was an old one that had evolved. 5,000 years. The same bacterium, evolving alongside us the entire time. Over those millennia, the bacterium acquired new weapons. A gene for surviving inside flea guts. A gene for spreading through lymph. By the time it reached medieval Europe, it had perfected its killing strategy. The final version. Fast forward to 1348. The Black Death arrives in Europe aboard merchant ships sailing from the east. Within 5 years, between 30 and 60% of the European population is dead. Entire villages were emptied. Cities lost half their inhabitants. Bodies accumulated faster than the living could bury them. The scale was beyond anything in recorded history. No war, no famine, no natural disaster has ever killed a larger proportion of the human population. The scale of death was beyond anything in recorded history. And for centuries, the question remained unanswered. Why did some people survive? The plague killed their neighbors, their families. But they lived. For decades, historians assumed it was random. Bad luck, good nutrition, geographic isolation. Then in 2022, a team led by Luis Barrero published the answer. It was written in the dead. The study was unprecedented. They extracted DNA from 516 individuals buried in London and Denmark. 3 groups: people who died before the plague arrived, people who died during it, and people who survived it and had children afterward. By comparing the genomes across all 3 groups, they could identify which genetic variants were under selection. They were looking for the genes that decided who lived and who died. They found 44 genes under the strongest natural selection ever measured in humans. The strongest signal came from a gene called ARAP2 on chromosome 5. People who carried 2 copies of the protective ARAP2 variant were roughly 40% more likely to survive the Black Death. 40%. That is the largest selection advantage ever documented in the human genome. In a single generation, the plague reshuffled the genetic deck of an entire continent. Before the plague, the protective variant existed in roughly 30% of the population. After the plague, it had jumped to nearly half. The mathematics of natural selection are brutal. A 40% survival advantage compounds across generations. Within 2 to 3 generations, the survivors' genes dominated. The genetic makeup of Europe was permanently altered. ARAP2 encodes a protein that trims pathogen fragments, so the immune system can recognize them faster. The protective variant produces a more efficient version. People with this variant mounted a faster immune response to Yersinia pestis. Their bodies recognized the invader sooner. The difference between life and death was a single gene variant you inherited from your parents. But the plague did not just select for one gene. 3 other loci showed significant selection signals: CTLA4 on chromosome 2, TICM2 on chromosome 5, and a locus on chromosome 18, all involved in immune recognition and inflammatory response. Together, these 4 genes created a coordinated immune defense. The survivors were not lucky. They were genetically equipped. But here is where the story becomes truly terrifying. The genes that saved them are now killing us. The same ARAP2 variant that protected against plague is today one of the strongest genetic risk factors for Crohn's disease. The immune system that evolved to kill bacteria faster now attacks the body's own tissues. The geographic pattern is precise. The populations that experience the strongest plague selection now have the highest rates of autoimmune disease. Northern Europeans, the descendants of Black Death survivors, carry an immune system calibrated for a pathogen that no longer exists. Crohn's disease affects roughly 3 million people in Europe and North America. The ERAP2 variant is its second strongest genetic risk factor. The same overactive immune response that once killed plague bacteria now attacks the intestinal lining. Evolution does not plan ahead. It solves the crisis in front of it and leaves the bill for later. And the bill has been compounding for 5,000 years. Each major plague outbreak selected for stronger inflammatory responses. The Neolithic plague selected for the first round of immune adaptations. The Justinianic plague of 541 CE killed an estimated 25 to 50 million people across the Byzantine Empire. It weakened Constantinople so severely that the empire never fully recovered. Ancient DNA from Justinianic plague victims confirmed the same bacterium, the same species, a different strain. Another round of selection, and the Black Death applied the strongest selection pressure of all 3 waves of the same evolutionary force, pushing human immunity in one direction. The 2022 study changed how scientists think about disease. Autoimmune conditions are not random malfunctions. The team tested it in the laboratory. They exposed modern human macrophages to Yersinia pestis. Cells from people carrying the protective ARAP2 variant killed the bacteria significantly faster. But those same cells also produced more inflammatory cytokines. The immune response was more powerful but less controlled. The same molecular switch that killed plague bacteria now triggers chronic inflammation in your gut. Crohn's disease, rheumatoid arthritis, ankylosing spondylitis, the immune system's overreaction to threats that are no longer there. Millions of people take immunosuppressant drugs today. They are chemically undoing what the Black Death biologically selected for. The scale is staggering. Over 300 million people worldwide live with autoimmune conditions that trace directly to plague selection. Your immune system is still fighting a war that ended 700 years ago. The East Smithfield Cemetery in London was opened in 1348 specifically for plague dead. The bodies were buried quickly, some in individual graves, some in mass trenches where they were stacked 5 deep. The excavation recovered over 600 individuals. Their teeth were intact. Their genomes were readable. From those teeth, the team extracted and sequenced 516 complete genomes, the largest ancient DNA study of a single historical event ever conducted. Each skeleton was a data point, each tooth a time capsule of someone who lost the genetic lottery. The DNA from the plague dead told one half of the story. The DNA from post-plague survivors told the other. The survivors did not just endure. They passed their advantage to every generation that followed. 20 generations. That is all it took for the plague to permanently alter the European genome. The signature is population-specific. East Asian and African populations, which experienced different plague histories, carry different frequencies of the same protective variants. The plague did not edit all of humanity equally. It edited each population according to its own exposure history. The population that rebuilt Europe after 1350 was genetically different from the population that existed before. Not culturally different, not just demographically smaller, genetically different at the molecular level. Every person of European descent alive today carries the genetic legacy of the Black Death. The plague did not just kill people, it edited the survivors. And we are still living in the edited version. 5,000 years of evolution, written in teeth, read in a laboratory, felt in every immune cell. The labs are still running. New teeth are being sequenced from new sites every month. Each new genome adds another page to the longest and most terrifying medical record in human history. Somewhere in the ground, in teeth not yet found, are answers to questions we have not yet asked. We open the teeth. We read the DNA. But the plague is still writing. If you want to know what else ancient pathogens have done to our DNA, subscribe. The sources are in the description. The most terrifying discovery in ancient DNA history is not that the plague existed. It is that it never left.